Good morning, everyone. We're going to begin. I just wanted to say a quick thank you to Simon Searchlight for shining a light on OCNDS and helping us accelerate the path to treatment. The Simon Searchlight team's dedication does not go unnoticed. They work tirelessly for our community to make our families' lives better, and we are forever grateful. After postponing our in-person conference twice, we are finally in the flesh. We are together. I am so excited to be here in person with you, and I am grateful to all of you families who braved airport travel, COVID, monkeypox, a car wreck to be here. I am Jennifer Sills, and I am the founder and president of the CSNK2A1 Foundation. And I want all of you to look around the room. I want you to look at each other. And I want you to know that you are part of something remarkable. You are part of a global family who is the driving force behind OCNDS research. When I was pregnant with my daughter, Jules, I used to think about what her life would be like, the college she would go to, the friends she would have, who she would marry, the robust life she would live. And little did I know that seemingly healthy babies at birth would have genetic conditions lurking in their genes that would change their lives forever. OCNDS was first described by Drs. Okur and Dr. Chung in 2016. And shortly after, my daughter was the 6th in the world to be diagnosed with OCNDS. And at that time, there was no foundation, no information, no website, no community, and no one was studying OCNDS. I asked our neurologist what we should do. She told me to contact the author on the paper, Dr. Wendy Chung. I emailed her immediately. I then typed up 2 pages of questions, and then I happened to Google her, and I found out that she was a powerhouse in the rare disease world. When we had our first appointment, we got through 4 questions, and she told me the cold, hard facts, which was no one was going to be studying OCNDS unless we started a foundation and funded research ourselves. She gave me an action plan to build a strong patient advocacy army. She told me to find 60 patients, have an in-person conference, create reagents, whatever those were, fund basic research, fundraise like crazy, and find researchers who will be champions for OCNDS. In 2018, we launched the CSNK2A1 Foundation. And that year, we made our first mouse model. We had our first in-person conference. We found over 60 patients, and we funded our first study. It is a fact that the path to treatment is slow and even slower in a pandemic. But we have come a long way since 2016 when OCNDS was first identified. And my wish is that you leave here today with great hope. I want to share some highlights since 2016. At the 2016 conference, besides Dr. Okur and Dr. Chung, we had one researcher, Dr. Heike Riepoltz, in attendance. At this conference, Simon Searchlight arranged for our first in-person scientific workshop. Throughout the weekend, virtually in person, we had 23 researchers attend. We have found our champions for OCNDS. Many of the researchers are still here, and I encourage you to speak with them. I encourage you to introduce your families to them. Don't be intimidated by the term researcher or scientist. They are brothers, they are sisters, they are mothers, they are fathers. They want to hear from you. They want to learn more about OCNDS, and you are the expert in how OCNDS shows up in your child. At this time, I actually would like to introduce the researchers that are still here. So I ask all of you to come up so we can introduce you to the crowd so people know who you are. So at the dinner tonight and at lunch, they can come meet you and join you, and you can learn more about them. And if any of you have ever been on our quarterly calls, you know that we start our calls and end our calls by introducing ourselves, saying where we're from, and giving one word about how you felt about how the kind of day went. And so, oh, this is awesome. We're spilling off the stage. There's still so many here. So that's the format that we're going to do here. I'd like to ask each one of you to say your name, where you're from, and a word describing how you're feeling about being here, about future research. Thank you.
Yeah, so I'm Ck2.
Oh, sorry, hold on.
And yes, I was the one who was saw some of you already in 2018. And I just repeat what I said then, that I've been working on CK2 for decades, and it was okay. But finding you, finding the community, finding the condition changed everything for me in doing research, because it gave huge sense, huge purpose, huge feeling of that my work is meaningful. And so we're all, I guess, super motivated to work on this, because yeah, it's just being part of the community on the other side, but in the end, we're on the same side somehow. And so I'm working in Paris at the Neuroscience and Psychiatric Institute. I'm working with the first mouse model and other upcoming mouse models to try to see what are the similarities, and can we use these mice to study potentially novel therapies to help OCNDS patients.
So my name is Jordan. I'm coming from Germany. I'm married, son of 16, and I hold a chair in pharmaceutical and medicinal chemistry at Münster University, and my group is actually developing methods or tools to evaluate the drug, how a drug is functioning on a certain target. And in this respect, we are working since quite a time on CK2, but direction care and the cancer drugs. And I came into the project initiated by Heike and by Carsten and by Isabel as well, which I knew before since a long time. I need to start it. But I guess I do not need to start again.
Yes.
I get you.
Now you can tell them your one word.
Yeah.
Your one word about how you're feeling.
I'm impressed about this meeting. I'm very excited, as well as Jennifer said for her in the beginning, to see all the people coming together, exchanging their experiences. But also, we had a researchers meeting yesterday, the day before yesterday in the morning, and we have seen the progress that we've made in the meantime, and are directing the work for the future. And just to mention for you, there are a number of publications, worldwide known journals about this group is coming out. And we always write and acknowledge the support of CSNK2A1 Foundation. But I said the next time you will write the name of Jennifer Sills in this issue, because she's the power, she's the motor behind this all, and it's very inspiring and inspiring to be here.
Yeah, I'm Alexandra Lee. So I'm a student of Joachim, so I'm also from Germany and Münster. So I don't have to talk about much what we are doing at the lab, since Joachim already talked about it. But I just want to say that it has been really inspiring to see all of you. So it's just to finally see the faces behind the thing that we work on every day has been really motivating me to push harder and work more on this. So thank you for inviting us here.
Good morning. My name is Carsten Niefend. I'm from Cologne in Germany, from the Institute of Biochemistry there at the University of Cologne. First, thank you for inviting us here, and I'm quite impressed how positive you are, and you organized all this in spite of something which is really not so easy for you. CSNK2A1 is
So you
the
don't
name-
feel so bad, even he has
is
trouble.
yeah, it is the name of a gene, but we normally use the name of the protein, CK2 alpha, and I'm working with CK2 alpha now since 30 years. And for a long time, we knew nothing about the genetic disease associated with this gene, and this all, of course, also gives the research a new perspective. And in our group, it is in particular Jennifer Hofstra here next to me who plans to do this research even more than myself. So of course, I hope that will be successful with what we are doing, and of course that this will also give some benefit to your community. Thank you.
Hello, as Carsten mentioned, I'm Jennifer Hofstra. I'm also working with CK2 alpha for more than a decade now. And I have two small daughters, 4 and 2 years old. And when we noticed that there's a condition associated with CK2 alpha, it was very moving for me, because at that time I had my first daughter and I was pregnant with a second one. And it really moved me very, very deeply, and still does. And I'm very happy to be here. And I do what I can to push things forward in the lab.
My name is Isabel Dominguez. I am originally from Spain, but I am working at Boston, Massachusetts, at the Boston University School of Medicine. And just echoing what everybody has said, it is really a privilege to be here and to meet you guys. Sorry, I'm gonna get up. It's really an honor, and I am so admiring of your resilience. Really, it's just terrific to see you guys. It's terrific to see you guys wanted to get to a solution for the disease and helping us help you. Anyway, so we are doing our best at Boston with mouse models and frog models of OCNDS that can help hopefully bring therapies. And again, it's really an honor and a privilege to meet all of you. Sorry.
Well, good morning also from my side. My name is Christian Werner, and I'm a student of Carsten Niefend in Cologne, Germany. And I'm rather new compared to the others in working with CK2 alpha for about 18 months only from now. And I just want to thank you for being part of this community and for the invitation to Jennifer Sills. It's quite emotional for me. Sometimes I cannot show this, but this is really something very special, and just wanted to say thank you. And I hope this will bring a lot of progress and a success for all of us.
Hi, everyone. My name is Lydia Bischoff. I'm a student at Boston University, and I work in the Dominguez lab with Dr. Dominguez, and I'm from Ohio. I've really been inspired since coming here, and I appreciate being invited and being allowed to come here. So thank you so much for sharing your experiences with us. Yeah.
Go up.
Hi, everyone. My name is Alexandra Lee, and I'm a student at Brown University. I work with Dr. Okur, and I also have a sister with OCNDS. So this research is so much more meaningful to me on a personal level. And I was also able to interview a few of you guys for a project that I worked on for school, studying the caregiver burden. And I hope that we will be able to explore this more in the coming years. But I just want to say that this experience for me has been I did not expect to do this. Really, really overwhelming in a really good way. I feel like a fire has been lit under my butt to I forgot to mention that I'm going to medical school next fall. So I hope that I can help educate my peers into becoming the best doctors for you guys. I hope that I can do everything that I can to support us and our family. So thank you.
Going out.
I need to go up? Okay.
Yes, yes, you do.
I think Jennifer, we talked about the t-shirt, large size. I don't think it's my size. I need an extra large. Well, my name is Halim. I'm a principal investigator at Sanford Research, South Dakota. So we just moved in basically this week, the new lab on Monday. So, and even with a big like road trip. So I said, I need to be here to meet you guys. I met some of you. I'm really grateful and honored to meet all of you here and looking forward basically to meet the rest of you. So we were working on small antenna called primary cilium. So we can think about your house as the cell and you have an antenna. So we found that CK2 is at the base of that antenna and it's basically tuning that small antenna. And so I hope that we find really great things about that. So yeah, I'm happy to be here. So thank you guys.
Right. Hi, everybody.
Take off your mask. Sorry.
Oh, okay. You can hear me though, right?
Yes.
Okay. Sorry, I didn't intend to be the last person. My wife just warned me that we were being called. I mean, I'm Volkan Okur. I'm a medical geneticist by training and also laboratory geneticist. I'm the guy who stepped on these people's shoulders on their works. I read a lot. I learned a lot from them. And I'm the guy whose life you changed, actually, not the other way around. And I'm trying to, you know, give the best out of me, like at least that I can be maybe accessible, even if like I'm lacking on some other fronts. So I'd love to be a bridge between you and these researchers, try to connect the basic research with the clinical findings phenotype, and try to listen any small issue that is coming up and not to ignore it right away, but try to be cautious about it. And hopefully I will continue to do this like to the end of my life. Thank you so much.
Thank you. Thanks. I had no idea the researchers, normally they're kind of quiet. So I am so happy that actually they said so much, and we, in order to run on time, I'll go over some of the other things, just foundation highlights that we have at our dinner. And at this time, you know, just so you know, in 2018, when we started, there was no parent advisory board. We didn't have 10 amazing parents that we have now that are on the board, who are working to have monthly calls with families, that are developing materials for families so they can at least ride a little bit easier on this journey, this OCNDS journey. So I'm going to introduce Terri Jordan, who's a member of our parent advisory board, to talk a little bit about the materials and the things that they're doing.
Thank you, Jennifer. Appreciate it. Hi, my name is Terri Jordan, and way back when in 2016, I heard that there were 5 girls diagnosed and my son. So this tells you how way back when it was very rare. I want to introduce everybody. First of all, I have a wonderful spouse named Jeff, and my older son is Logan. And then Cody, who is now 20 and will be 21 at the end of this month, is the one diagnosed with OCNDS. Whoops, sorry. All right, understand that OCNDS does not affect everybody in the United States. It is a global genetic condition, and our families do expand the globe. OCNDS, we speak more than 9 languages. And what is exciting about this 9 languages is together we're stronger. And we encourage every family virtually that is joining us and who's in person today to join us for our family Zoom calls. We have the technology using Wordly that we can do translations that are happening so that you are connecting with families instantly and you can ask your questions that are happening to your child at that moment. We always, as Jennifer asked all the scientists, we ask the families, what does this call mean to you? And these were the common words that are used through every situation on our calls. It's relief. I finally feel connected. I feel hopeful for the first time. I feel grateful, excited, understood, not alone, that there's others in the same situation, supported. And the biggest one, I have now found my family. The Parent Advisory Board has been busy. In our first year, we put together our brochure, What is OCNDS? And the minute you rattle that off, doctors look at you with that deer in the headlights look, family members look at you and they're that, or if you say CSNK2A1, they're like, I'm sure I've heard of it. Right. Okay. So we have a brochure put together. You can download that brochure from the foundation's website. For those of you who are joining us virtually, we encourage you to print it out and give it to family members, give it to teachers, give it to your doctors. But if you are here and you want to pick up additional copies, please do. We have them over there on the table. The Parent Advisory Board sends out surveys. We ask that you do participate in these surveys. We did this year the COVID survey and we did the Pulse survey. This is a check on the community. We want to make sure that everybody is okay. We want to make sure that if somebody is needing some extra supports or information that we are able to get that out to you. We don't want you to feel alone. Our latest piece is the CSNK2A1 awareness card, also known as a TSA card. This card is fill in your family member's name and it explains what is CSNK2A1. Who can you give this card to? You can give it to a teacher, special education providers. I think that is very important for them to have. When getting a vaccine or a blood draw, give it to doctors. Like if you drop your child off at camp, strangers who want to know more that might be stopping you in the middle of the grocery store, and extended family members so that they can understand what is happening with your child. Ways to participate. We encourage families to participate, and this is how you can make us stronger. We're going to start with join the OCNDS family Facebook page or the friends and family if you are extended family. The Instagram, Milestone Mondays, cut off there. Milestone Mondays, we want to hear about your child's or family member's success. This may be, I posted when my son graduated from high school, his picture. I want to hear about that your child finally took their first step at age 4. I want to celebrate with you. If your child started speaking at age 11 and was able to say, I love you, I want to be there going, yes, because it happened for you. So this is the one way you can help your family members celebrate with you is by putting out Milestone Mondays and participating that way. The My Story, you need to tell us your story. What did you do to get to this point? What things did you have to do? And my story is when I got my child home, he couldn't swallow. So for him to be able to eat, it took 45 minutes of stroking his throat to be able to swallow. That is part of our story. It is part of our journey. And it's part of what made him successful. And he continues to be successful every day. We need to hear your journey and your story so that we can inspire other parents and let them know they're not alone. Rare disease truths, they happen. Things happen, we share them. And some days you just go, I can't believe somebody said that to me. And we need to share that. And it also is a way to help arm our parents and family members in this group to go, okay, I don't have to take this as a personal affront. I need to educate this person who said this information and be able to handle it. That's the important part. We need to educate the community around us. Simon's Searchlight, we are encourage everybody to participate in that program and do the medical research side of it. We need to get 100 families on board so that we can have this group move forward to the next level. So this is what we need to do next. Thank you for being here in person. Thank you for joining us virtually today or in the future. We appreciate it so much, and I am so glad to be a part of this OCNDS family and community. Thank you.
I'm supposed to introduce you, but I don't think that you Dr. Okur, Okur developmental syndrome.
Right. Yeah, you can hear me. Okay, great. So yeah, today I'm going to do something different. I mean, not totally different, but I'm not going to delve into the clinical aspects that much. I'm going to try to give you some updates on where we are in terms of some clinical research and also some bullet points for the basic research going. And hopefully then I will be able to move you to also register for Simon's VIP. Okay, how is this going? Oh. Okay, so something that you will have questions about, that so far we have 19 publications, and those are clinical publications. And in those publications, 51 individuals reported to have CSNK2A1 variants, which sounds very low, I know, and there's a reason for that. But like even lower than that number is only 36 individuals were described with clinical and mutational data. So the reason that this number is low than what we know by the foundation's number registered is that we are only allowed to put the published number there. So that's why these natural history studies are very important, to increase that number and to let everybody else know that it's not just 50, it's not that ultra rare. We have like more now, up to 200 individuals. But still, maybe thanks to the robustness of those papers, we were both first attempted like a year ago, and then we were invited again to write the gene reviews chapter, which is a reference textbook for clinical follow-up management issues. And we completed the process. It took about like 6 to 8 months. And then now we are online since June. And there we kind of grouped the most commonly seen findings here on the table, which is pretty much actually, more or less in line with what we observe from the larger group and also from the preliminary Simon's data. And we also put there like some management recommendations, but those are not OCNDS specific per se, actually. Those are like the generic recommendations that are used or that are written by the federal guidelines for individuals with some challenges. So if, I know most of you have some struggles to access to some resources, some educational programs, and the physicians in your local area where you're living, there might be some, resistance or disbelief, maybe because of also the relatively milder spectrum. You can refer them to this page. And I mean, I know at least worldwide, this chapter is accepted as a reference textbook. So what else are we doing right now? The first and the most important one is the natural history studies. So, and along with that, if there is any difference between different variants which we call genotype-phenotype associations. And those natural history studies are important because many of the studies, published studies, we only do a cross-sectional, just one at a time finding. We don't know the development, we don't know the longitudinal progress of your lives. And also, especially when attempting to do genotype phenotype studies, it is really important to account for the age of intervention starting, when the speech therapy, occupational therapy started. So otherwise we might have some false pretense or like give some false hopes to families. But one thing is for sure, as early as the therapy is started, it is great. Other than that, recently in a few years, in the last few years, we are getting emails from both physicians and families that they have a different type of variants that are reported or described, and those are deletion variants. So I'm going to explain what is the difference, but we have gotten in touch with some of you and talked with you and invited you to the study to also delineate and describe the findings of those deletions and deletions of CSNK2A1, and that they are also associated with neurodevelopmental issues. So the larger community will also be aware of that, and it's also gonna kind of guide our research projects when we look at the functional studies. And the other three is the facial gestalt. So different than the other past genetic syndromes, there's no striking facial features, but as you know that we know each other very well, and there is something, it might not help someone outside maybe United States or developed countries to order targeted testing for this gene only, but it might help us to classify VUS as well when if we don't have any right away functional studies to classify that one. And other than that, I know many of you are asking about adulthood, and since we don't have many older individuals, we are still learning about it, but we have one resource right now that we want to look into it. It is from UK Biobank. They have sequenced like thousands of millions of individuals, and there might be some individuals who are carrying variants in this gene and who are now in their adulthood age, and we want to look at what presentations they had at the time of the enrollment to this study. So, and maybe it might also guide us for future. And then there's outlier symptoms that, like I said, like we are also taking those seriously. Again, like we need to be cautious about to link those symptoms directly to the OCNDS, but when we look at the genes and proteins function, they make sense, but we need more data. So on the back end, it's not maybe going globally on the Facebook or other avenues, but I'm in touch with individual families for immunologic profile studies when there is like ataxia, some encephalopathy issues. So we are taking those seriously as well. But we are not going to make a larger announcement until we find any association or not. So a little bit about clinical deletion variant study. So these are the variant types that I tried to simplify. The normal word is the one big fly had one red eye, and these are the representations of what the variants are. So the most common ones are missense at the top that most of you have, and here at the bottom there's like crowded numbers. Those are deletions that the entire gene is deleted. So what we are saying is these are also causing this problem. And now we have like this in this representation, like at the top we have 10 individuals, and it's going to be more. And we also have other individuals in other resources that will support our case. And here is the CSNK2A1 gene, and this gene is the most important among all the other ones surrounding it. So I'm going to now give a little bit update on the experimental and why actually we are doing this, because I know it's going slowly and it might sound frustrating as well, like in some clinical areas. And the reason is we need to understand how those variants affect the biological process. So on the left-hand side, that is a tree that this genes like protein, the CK2, the major protein is a member, which is a kinase in a large family. Our gene is on one of its branches. And what these kinases usually do is like they usually regulate other genes like a dimmer, like this, and they either increase the other genes like functions, expressions, or decrease them. And that is important. And it's also important depending on the function of the downstream gene. And this like technical slide is not to confuse you. It's just like this protein per se, it has a role in many biological processes. So that's why it's really important to figure out what is the driving cause and what we should target when we attempt to fix and or correct. So like all those researchers said that, sorry. So and what we are looking is that first, if there is any change in the protein's amount, is there a decrease in produced protein? So which is going to be the case for example, deletion variants. And the other one is mainly for the missense variants. There are like a few possibilities. One is the protein might still lose its function and cannot regulate those other genes that I mentioned, or it might regulate its normal targets, but more than necessary, which those are the technical terms. I'm going to give the slides later. So if you hear those again somewhere else, you can reference it. And the other one is the protein regulates different genes than its native targets. So we are trying to figure out what is the role of those variants among these possibilities. And those loss of function sometimes, like they can also behave like haploid insufficiency, but it might also be, again, the degree might be lower than the entire loss of the protein. There might be some confusion, and also there's some like interchangeable terms. And the other one is the protein structure. And this is like how those variants, like now, again, thanks to the researchers' previous work, we have a knowledge about the 3D structure of this protein, and we want to see like how the variants are affecting this 3D structure, which would then affect their interaction with other proteins, because everything is going on 3D level in the cellular level as well. And this one important thing is like how this affects our sibling gene, CSNK2B, and also other genes. Because as you may have heard, that there's also another condition associated with this other gene, which have like some similar findings, also some different findings, which is important as well. So we need to figure out the similarities and differences. And again, I'm going to skip this quickly. These are going to tell us like what variants are going to act in what way. And how are we doing this is first, like the animal studies, we use animal studies, and it's to show if the animal samples, they show the same or even similar phenotype as observed in humans, because there's going to be differences anyway between animals and humans. And what are the biological changes in their tissues, and particularly brain, because it's really difficult to access the brain. And in laboratory studies, we are tried to test the variance function, again, to classify like in what way they perform, again, change of substrate specificity, different targets, and less function on the native targets, etc. And also in structural analysis, that how the structure of the protein changes with a variant at a given position, for example, for this variant, the most common one, and how it's going to affect the further downstream researches. And the reason to do this is, for many variants, like, for example, we know right away, we don't, like the K198R, for example, or R47, they are right away pathogenic variants because of the clinical data. But we need to understand, how and in what way they are doing this. And also these studies are more feasible. And also with particularly animal studies, we need a baseline benchmark. So what are we going to attempt to fix? So it might be just, okay, we might, identify something, but then, so we wouldn't know if we don't know the baseline problems in those mice with the particular variant. So what are we going to change? What effects that we will say that, okay, this is working or this is not working? And so there were a lot of studies with this gene, and like I said, I read a lot, I learned a lot. They did the major work. The major one on the animal part is the deleting one or both copies of the gene. There were some differences between mice and humans in terms of that. But Dr. Dominguez worked, I guess, that one, deleting the copies. But we are still going to look at those maybe in more detail. And post OCNDS description, there's like targeted variant carrying mice, the common variants, and Dr. Repholz is working with them. And she showed that there are some behavioral tests showed similar to behavioral problems that we observed in our kids. And also she's also doing further functional studies on those. Dr. Dominguez still working with frog studies, particularly to, elucidate the anatomical aspects, like, or how the embryonic development is affected by the disruption of this gene. Again, on the laboratory side, Dr. Dominguez, in this study, I mean, she's the author, but Dr. Repholz as well, and I guess others as well, like they reported that certain variants are losing the degree of their kinase activity, loss of function per se. But then like this year, 2 other papers came. The one is like Keffer et al. They reported a change in those altered substrate specificity, which actually is not totally negating the Dominguez et al.'s findings, but it is also, expected finding that if there's a change, there will also be a loss on one aspect. So these are all good studies leading to our final hypotheses. And there is also now other structural analysis studies by Werner et al. All of these researchers are in this room right now. I'm just using the published citation style. And they also showed that with those structural analyses that there is a change in the 3D structure, which also might result in target recognition. So at the end, it might look like that so the X and Y probably are the normal wild type, the unmutated CSNK2A1's target. And then if there's a missense variant, there might be some additional variants like Z amount of genes. And also there might be a change in the degree of Y amount of genes phosphorylation status. So they are still looking for it. And hopefully, but I mean, we have come a long way. It's, in my opinion, it's really going great. And we will have like a more robust results soon. And what we need to do going into the future is we need to continue doing these studies. Again, now we know, like at least on the gross level, that we see some very similarities, but we need to define, map those phenotypes, both on the clinical level and also cellular level, especially animal studies. Again, like the reason is what to correct. We have to know about that. And continue doing laboratory analysis. This is gonna help us, especially both, understand the downstream target, either changed or like decreased, and also classify novel variants. I know like we have very common variants, but we also see like individual variants that are maybe not in the regions of interest or the most commonly mutated regions. So we need to, have more robust techniques to classify those as well, because they're going to be needed to be classified as likely pathogenic. And also continued structural analysis, again, also to understand the ways to access the abnormal protein on the 3D level, how it changes. And it will also help us like to identify maybe like some targets that might interact with that 3D, like change 3D structure, because these are really very like, delicate technical, like a 3D mapping things, like very small spaces that one reagent, one drug that might go in there or not go in there, even if maybe it has a normal interaction with the normal protein, it might not have with a variant, the mutated one. So it's really crucial to also identify those. And one other thing that is, I think, the high priority is that we need to see the global picture. So it's really difficult to do the functional studies one by one, like with every, known or identified target. So we need to look at the global picture, and to do it, hopefully, we're gonna be able to use the iPS cells, the stem cells, and then differentiate them into brain cells, hopefully, and see like how the phosphoproteomic profile, like I said, like dimming profiles is changed between the native and in the like variant carrier cells. And this is not an actual data, by the way, it's just a representation. So the change will be like this, some white areas are going to become green or red, and some reds are going to become green or white. I guess that's all. And I'm not very good at this, but I would like to thank again Dr. Chung and all those researchers again. I'm trying to be a bridge, but it's your work. You have done a lot. I hope like I can be at at the same help that I am trying to be to the families when interpreting the research findings, and Alexandra as well, especially with the deletion variant study. And I mean, I can't thank anybody else more than you. Like I said, I learned a lot from you. I keep learning from you, then making new ideas, thinking about it. And I really appreciate it. You made me an expert in one disease, like around the world. And I'm trying to give everything back to you. Yeah. Thank you.
The gene reviews that Dr. Okur mentioned in his talk, in the packet that you received at check-in, it is in there. And I encourage you, if you need more copies, I'm happy to give them to you. It's finally, they are not necessarily the standard of care guidelines that we're looking for when we finally have the 100 families who join Simon's Searchlight, which Wendy, Chung, and Dr. Okur will finally write care guidelines because care varies wildly from patient to patient. We have some patients who have speech delay and they receive speech once every 6 months, and others that receive it 5 times a week. And we would like to make sure our kids have the best chance possible and are really receiving the services they need. The gene review is one way, and it's a beginning step to show your doctors this and show your school district, if you're fighting for services, your insurance company, to help get the services that you need for your children. So just a little update. And for those that are online, all the publications that Dr. Okur mentioned together with the gene review are available on our website. Sorry. And then now the esteemed Dr. Isabel Dominguez from Boston University. We are very lucky because in this room, including Dr. Dominguez, was working on the protein created by our gene CSNK2A1, Ck2 alpha for years and years. They wrote the book on it. And so we're so lucky to collaborate with her and to have her here today to tell us a little bit more about the gene, the protein, how it works, and to break down a little bit when you're hearing like missense variant, and to understand a little bit more about what's going on in your child, so you can be armed with a little bit more information when you go to your doctors and explain kind of what's going on. When you look at these numbers and you say, it says 198R, what is that? Where is that? And so she's going to give you a little bit more detail on the gene. Thank you.
Thank you, Jennifer, for inviting me to give this presentation, and I'm hoping that is good enough for all of you to understand. And if not, just let me know, stop me at any point, and then I'll repeat or I'll change it, and we can talk afterwards also if you would like to hear more. All right, so we're going to start with the basics. What do we know about Ck2 alpha? And as Volkan said earlier, Ck2 alpha, sorry, Karsten actually mentioned that Ck2 alpha is actually the protein that we work with. So we're going to start with the basics. So CSNK2A1 is a gene. And what we mean by a gene is just a blueprint. You all understand what a blueprint is. So it's just a blueprint. Just think about it as that. And what that blueprint help us do is to create this Ck2 alpha protein. That is actually what works in our organism. All right. So again, the blueprint is there. We use it to create the protein. Okay. So this is what we know already about Ck2 alpha. Again, Ck2 alpha is what works. We know how large it is. It is 391 building blocks. So like this, from 1 to 391. And those building blocks, we call them amino acid residues. So when you hear the word amino acid, all it means is one of these blocks. All right, this is our jargon. You don't need to learn our jargon, but you need to learn that these are the number of blocks. We know also how it looks in 3D structure. And again, with this, what we call structure, and Karsten and Jenny and their groups have done a tremendous amount of work in how it looks. And that's how it looks in 3D structure, like anything. So it has a three-dimensional structure. In my presentation, in many cases, I have represented it like this to simplify this this beautiful 3D structure. We also know that it's always working in our bodies. It's like if you think about your house, the refrigerator is always working because it always has to keep the low temperature for your food. However, there are other proteins that we have in our bodies that we only switch them on when we need them, like the TV in your house. You only switch it on when you want to see TV. But the refrigerator is always working. So think about CK2 about something that is always working in your body. Then we know also that it can work alone, all by itself like this, but it also can work together with this other protein called CK2 beta that Volkan was just mentioning, that it also gives rise to another disease that is called bovines. And they form what we call a tetramer. Again, tetra for 4, mer for number of units. So again, it looks like this: a CK2 alpha, another CK2 alpha, a CK2 beta, and another CK2 beta. And that's how it looks in 3-dimensional structure. And again, I will represent it like this to simplify this beautiful structure. So again, alone is absolutely fine, but also works with other proteins like CK2 beta. Yeah, is that clear so far? Yeah, very good. So next slide. What else do we know about CK2? Oops, I think I went too far. All right. So we know that that it affects us other proteins. As Volkan was saying, this CK2 alpha is part of a large family of proteins that are called kinases. Again, it's one of these names that we use for proteins. You don't need to remember it, but what you need to remember is what Volkan said, that what CK2 does is takes other proteins and change them in a way that these proteins now do something different. Either they work more, or they work less, or they go to other places and do other things that they haven't done before. All right, that's all it does. It changes other proteins. And if you wanted to know what it is, it's everywhere. And this is the beauty of this, this protein, that it's in yeast that make your bread, it's in plants that we eat in our salads, it's in fruit flies, it's in mice, it's in frogs, and it's in fish. And what is important from this is it allows us researchers to use all of these, what we call animal models, or this is not an animal, or this is a plant actually, but all of these different models to study CK2. And you saw earlier from all of us that we were here, we are working in mice, like Heike. We are working in frogs in my laboratory. We have the people in TGen working in the fish. And we also have researchers doing fly studies that were not here in this presentation. So anyway, so we can use all of these tools to help you. All right, this is the bottom line from this. Is that clear? Yeah. So that's how it works in a nutshell. And what I'm going to show you now is how is it made. Yeah, how is CK2 made? All right. So again, I want you to think about what I said earlier about the blueprint. So the CK2 gene is the blueprint, and we are used to see blueprints from how to construct a TV or a computer. You see those constantly, or a car. This is the blueprint from CK2, and it's just a bunch of letters. That's what it is. That's how it looks like. Again, you don't need to remember any of this, the same as I don't need to remember a blueprint for a TV. I just need to know how to use the TV. So, but if I were a worker that were constructing a TV, I will need to know that blueprint. And we're going to talk about the maker of CK2 alpha. So anyway, and again, this is what works. And again, it's those 139 blocks that we call amino acids that make this beautiful 3D structure. All right, so going to the next slide. This that you see here is called the ribosome. And again, you don't need to remember that terminology. But what you need to know is that the structure knows how to read that blueprint and make this protein. And this is amazing because since the start of life in Earth, that structure has known how to do this. And it took humans all this time until last century to discover how this teeny tiny structure was able to understand this blueprint to make this. So again, it was researchers last century, and again, they work, of course, they were awarded the Nobel Prize for this work, and it really opened the work of genetics, and that's how we can know now the mutations that we all have in our bodies. So again, if you look at the blueprint for CK2, it has 1,000, sorry, my numbers It takes a long time to read numbers in English when you are in when you are speaking of another language. So forgive me for my numbers if I made a mistake. 1,176 position, 76 positions, 76 letters that we call nucleotides, each one of these letters, and they are A, T, G, and C. And you will see this in your, when you get your, how do you say, your specs, you will see some of these letters in there again. So it's a mix of all of these letters. And again, what this ribosome is able to do is able to read these, and every 3 letters, what we call a codon, it makes a block. All right, is that clear? So from this long blueprint, every 3 letters makes one of these blocks. And let me see. And what the researchers last century were able to get through is what is the code that the ribosome uses to interpret this blueprint to make this beautiful protein. And this is what we call the genetic code. And if you have seen this before, you know that we have again 3-letter groups, and all of the ones that start with T are here, all of the ones that start with C are here, all of the ones that start with A are here, and all of the ones that start with G are here. And then the second letter, all of the ones that have T as the second letter are in this column. Then in this next column, all of the ones that have the C as a second letter. Here, the ones that have the A as a second letter, and in the last column, the ones that have the G as a second letter. Yep. Do you see that in the, and then again, so for every one of these, then there is T, C, A, or G as the last letter. Yep. Is that clear? How is read? I'm hoping that this is okay. Yeah, so far. Yep, good. So we're gonna see the first 2 blocks and the last block for CK2. So as an example. All right. Let me see. So the first 3 letters that you can see there are A, T, G. And we find the ATG right in here in the A row, ATG, and it means methionine. So that is the block, it's called methionine. Again, you don't need to remember the names, but it's just that this ribosome interprets that position 1, 2, 3 ATG as methionine. Then it goes to the next block, the ribosome, and reads apologies. Okay, it reads TCG. Can you find TCG in here? So T in the first row, is that right? Yep. C in here, is that right? And then G, is that right? Right in there. Yep. Cool. So you can read the genetic code also. So there it is. So TCG, and it makes another block that is called setting. Again, you don't need to remember any of these names. So now it has the first block, and now it puts the second one. And the ribosome continues reading and reading and reading and reading every 3 letters and putting a new block until it hits the end, and the end is positions 1,174 to 76. It reads this, that is TAA, and it's right here, TAA, and those 3 letters tell the ribosome, stop, we are done. That's it. We are finished with all of the blocks. And this is it. So this is all of the blocks will be done, and you'll have this structure that eventually will become the beautiful 3D structure for CK2. Is this okay? How CK2 is made? Yep. So now we're going to go to the next set, and we are going to talk about the CK2 variants so that you can see what that you can understand, I guess, how to read when you get your informes or your informes translate. Thank you. Sorry, sometimes when the English doesn't come, it does not come. So, and there is nothing that I can do for it. So apologies for that. So anyway, so again, we were talking about before about the CK2 protein and all of these 391 blocks. And when you have a CSNK2A1 variant, it means that the blueprint has somehow changed. Yep. And then what it means is that one of these amino acids, one of those blocks, has changed now. And what it means is that the protein works differently. And what Volkan was telling us earlier, that it can work in many different ways. And we don't know if it is more active, less active, if it goes different places. We don't know yet. If it interacts with other proteins differently, we don't know yet. And this is what we are trying to figure out. But in any way, so there is one of the blocks has changed. And I'm going to talk only about what we call missense mutations. That is what, again, as Volkan said, the majority of the patients have, but we can talk about the other ones during lunch or later on during the day if you guys want. Let me see. So this is, this is again the structure of CK2 for the first block 1 to 391. And this is the number of patients that had been found for some of the major missense mutations. So there is the K198R, for example, is here. Then is the block 47, the block 50, the block 156, 160, 191. All of these blocks are the ones that have the most patients. Yep. And let me see. So we are going to take, I think, the example from 198 and 47, I think, to show you. And again, you don't need to remember any of these. This is not important. But what is important is that we know which blocks are affected. And then we go to the next slide. And this is the first example is, of course, for K198R. And this is what you will see in your genetic report. In your genetic report, you will see a C, and then 5 is 593. I cannot read from here really well, but A to G. C, what it means is the gene itself. That's all it means. So that's, you think about C as the gene. All of this, remember the blueprint, all of the different letters, the 1,176 letters. And all it means that in position 593, originally there was an A, but now there is a G. That's all it means. Yeah, understood? Cool. And then this P means protein. So we went from the blueprint, the DNA, the gene, to the protein P, all right? C to P. And what it means is that in this, the codons, the 3-letter codon, where 593 is, will make for this block that is called lysine. Again, you don't need to remember the names. And lysine will be in position 198 within the protein. So this is actually the position. This is the start of the protein right there. And this is the position for the block 198. And this is how we abbreviate it also. These blocks have 3 names, one full name, one shorter name, and one nickname that is just a single letter like K. So the lysine, that's how it's called. It's also called LIS, and it's also called K. Again, you don't need to remember all this terminology. This is our jargon, and I'm sorry for this. It makes it too complicated for everybody else to understand. We scientists are like this. Anyway, so the codon for this lysine was AAA, and you can see here the change. So this A was changed to a G, as it says here, from A to G. And again, that changed this lysine here from a different block that is called, again, arginine or ARG or R. Yep, is that clear? Yep. And then I'm going to show you, I think, the 47. I'm still on time, is that right? I'm still on time? Okay, sorry. Sorry, I'm going to show you the 47. Again, same thing. 47 is coded by positions in the blueprint, 139, 140, and 141. All right. And the 3 letters that make that block, that is an arginine block, are C, G, and A. And then we have 2 different mutations here that we have put. The one that is a C, this C, the first C, sorry, this is fading, but the first C for a G, or in the next one is the G in the middle is changed for an A in here. And when the ribosome in your bodies reads this, instead of reading this, it's going to read this or this. Instead of making an arginine, this block is going to make a glycine or a glutamine. Yep. And then these are the shorter names that we use for them, GLI or G and GLN or Q. And again, this is position 47. This is the start again. And this is the protein goes like this, and then goes like this, and then boom, that is 47 right there. Yep, this is where the block is. And all right, I have, I guess, another example. I think this is the last one, sorry. Is this clear? Do I need to go through this example also, or can we skip it? What do you guys want to do? Let's skip it. Yeah, we learn everything already. Cool, fantastic. You guys are cool and smart. Let me see. Okay, I have also this other one, so you guys want to see those. And this is my last slide. And I just wanted to remind you what we talk about, the blueprint, that is the gene that is formed with those A, T, C, G letters that the ribosome reads in groups of 3 and makes one of the blocks that we call amino acid residues, and makes the product that we, oops, sorry, apologies, there's some more of them, that are these 391 blocks together. We talk about the variants, particularly the ones that change amino acids, like the missense mutations. Missense is just means that it's not the same sense as it was before. And I really would like to thank you guys for all your help. Without you, we would not be able to help you. So you giving your data away, like which mutation you have, helps us understand the disease, help us understand what we need to do and how we need to prioritize our research. And I cannot, I mean, again, I'm gonna tear up again, sorry. And also, of course, to Jennifer and the CSNK2A1 Foundation that you are part of, because it is a fantastic support group, is supporting us also and bringing all of us as a family also. We have meetings regularly to share our data and to think about what we can go next to help you guys. And Jennifer is the glue that binds all of us together to help you guys. Sorry, I'm gonna tear up again. So again, thank you to all of you.
Isabel, it's super informative because I know that when anyone's trying to join the Facebook group, we're asking, what's your mutation? And everyone's like, it says CSNK2A1 or it says OCNDS on my report. And there's a reason why we need to know what the variant is. And as you can see in this case, it's almost a case of bad luck. We have 3 billion, letters that make up our entire, genome. And in this instance, it's 1 letter for most people that have OCNDS, 1 letter, a 1-letter change that are causing all these symptoms and changing our loved ones' lives. And so it is quite remarkable how important this gene must be in development. So thank you for learning more about kind of your mutation today. Sorry, and Wendy, are you next? Okay. Again, someone I don't need to introduce, the famous Dr. Wendy Chung in the flesh. And then for those of you on the screen. Thank you.
Now we're on. Excellent. Okay, so good to see everyone here today. And for those of you at home or watching later, all of this is going to be posted. So feel free to take pictures if you want to, if there's a particular slide that captures your imagination, but we're also going to have all of this posted. So if there's anything that you feel that you want, don't feel like you furiously have to take notes. I know others have said it, but I just want to tell you, you've got a huge team of people behind the scenes. Jennifer is tremendous. She's been with the organization setting everything up. So I just want to acknowledge, I know life isn't easy for any of you, and to do on top of that, being able to carry the community organizationally. Jennifer literally flies around the world, as many of you know, to be able to bring in new families into the group. And Jennifer, I just want to thank you personally. You're an incredible asset to the group. And as Isabel and others have said, you guys have a tremendous team of scientists behind you. And so at the end, I'm going to just allude to what I thought was a lot of exciting work that was discussed on Thursday. But just to say, I can palpably feel the field moving forward, and it's because of this partnership. So Today, I'm going to talk about some of the things that I said in the main room, and I'll make those comments relatively brief because I think most people heard about them. But the point that I want to make is that Isabel was starting to, or in fact said this very, very nicely. There is a lot of differences within this group, and within this, it's really important that we have everyone be able to have themselves represented because I don't want to leave anyone behind. And there are enough differences based on those A's, T's, G's, and C's that Isabel talked about, but also in terms of the variability, differences across the life spectrum, differences by where you are in the world, lots of differences, and we can learn from each other. I'm convinced that some people have figured some things out, and we want to make sure that we disseminate those things that folks have learned. And so at Simon Searchlight, that's what we're trying to do, is aggregate that so it makes it easier for you as families, it makes it easier for researchers, that we can really move things forward. And I'm convinced that we can do it even better and faster than we have been. With this, it takes an incredible team behind the scenes, some of whom you've met here, but just to let you know, it really is an incredible team in terms of powering all of this, everything from neuroscientists to bioinformaticians to psychologists to physicians, a huge team of people that are all right step with you behind the scenes trying to make this work. And we're committed to doing this. We've been doing this for 10 years and counting, and we're going to keep on keeping on with this. So with this, like I said, you've got lots of people being able to help you along the journey. With this, we want to hear from you, if not today, at any of the breaks in terms of showing you what we know. There's a lot that we don't know. So either as researchers, as families, if there are gaps in terms of what we're doing, please tell us. We want to make this better for you. And we've continued to iterate since we started, and we'll continue doing so as we go on. Again, I think you all know, but there are folks outside here in the lobby so that if you either don't know if you signed up, you couldn't remember if you signed up for that study, you're not sure if you finished things, you're stuck, you can't remember your password, any of those things, the folks out here in the lobby will help get you on track and make sure that you can sign up if you haven't already. And I will give a lot of credit to the research team. They've been really flexible this weekend. And so some of you who have at the last minute decided you wanted to change your times or sign up for something, we're trying our best to accommodate everyone. So as you're doing it, if there's something you want to do, we will try to squeeze everyone in. We're making extra slots literally as we speak. In doing so, just as Isabel said, it's really important that we see your genetic test report for all the reasons that she mentioned. We want to verify and make sure that you're in the right club. Thankfully, most of you are in the right club, but I've, over the Searchlight career, found people who weren't in the right club and gotten them to the right club. And so it's helpful for you yourselves just to make sure you're on the right journey and you're on the right trail. As we're doing this, we are trying to make, as I said, this internationally accessible, all languages, all time zones, having staff that literally are around the world to be able to help support you. And it is not one and done. So let me make that very explicit. We need to understand how things change over the life course. And so we want to keep following your individuals, your children, your family members, so that we can understand how things are changing, what's working and what's not working. And so again, tell us this either just literally by sending an email into the on the upper right-hand section of the website, send an email to us or be able to fill this out in your questionnaires. Okay. So as we're going through, we have been trying to make this more accessible to families, both with your individual reports. So if you fill out certain of the questionnaires to be able to get information you can take back to your providers, as well as what we're learning as a group. And again, if you give us feedback about what's working or not working, we'll try and improve this to make it more understandable. The researchers already know this, but for those of you who have been giving blood samples upstairs, thank you, thank you, thank you. The researchers are using these because we're taking these blood samples and making them into brain cells, believe it or not. And I know that sounds kind of wacky and unusual, but we are doing it. And the Simons Foundation, along with the Nancy Laurie Marks Foundation, is underwriting the cost of this. And so making this de-identified, but available to people, to researchers around the world in terms of being able to do some really sort of fundamental neuroscience to be able to understand treatments and test out different treatments in a dish before we get to the point of testing out treatments in a person. We want to make sure that those things would be doing what they think they should be doing. So within this, I'm going to switch gears now and really focus on what we've been learning through the OCNDS registry. So within this, I just want to show you the numbers. I think the numbers are great, but as I was saying, I gave Jennifer a charge, and I know it's kind of a scary number for her, but I said, let's see if we can get first to 100 members in the community, and then let's see if we can get to 200 members in the community around the world. I know we're out there, like we know by the count, they're out there. And with that, I hope we'll be able to get to standards of care, and with that, be able to help to advocate for better coverage for certain services, certain therapies around the world. And it's a big world. And so as we're doing this, we do need this to be evidence-based. Right now, what's our count? Well, we're at 83, so we're close to 100. So if we can go home and phone a friend or tap a friend on Facebook, not coercively, but be able to do this in terms of inviting them, I'm pretty sure we can get to 100 by the end of the year for sure. As we're doing this, as Isabel said, we're looking at the genetic test report. And with this, we're not quite at 100 there. So that's kind of the first thing to make sure that everyone's in the right group. And then we're flowing downstream from that. And so we want to make sure anyone, again, who's stuck within this steps here, these first 3 steps, which are kind of critical to everything, we're going to try and help people get unstuck as we're going through this journey. With this, I will say there's an incredibly important scientific question that's coming up, and it came up in the group on Thursday. And so in particular, these are, as Isabel was saying, all the different types of genetic variants. And I'm not going into all the detail because Isabel did a great job with that. But I will point out that there are a few types of variants that we consider to be ones that make it such that instead of having 2 copies of the protein showing up and doing the job, one of those copies is not showing up and doing the job. So it's having half the amount of protein that's out there. That ends up being scientifically a very important question to understand. If you only have half the amount of protein, is that going to be good enough? Or do we need to be able to have closer to 100% of the protein or something like 90%? So if there are individuals in the room, for instance, and again, if you're not sure and you'd like any of us to look at your genetic test report, but have these particular types of variants that we call frame shift, nonsense, or deletion, that essentially take out half the protein, that you end up being incredibly valuable to understand certain therapeutic strategies we might try. And so your sort of donation in terms of understanding and getting to understand whether or not this is an issue or what the issues are, help all of these other folks in terms of thinking about, do we need to take away the protein? So for all of these folks over here, if we take away the protein, will we still be okay? Or will we be left with still problems that we have to address? So that's a high priority item that came out from the scientific group. I won't go into this again because Isabel did a great job, but that's just showing where the genetic variations are. Let me just say one thing. And as Isabel was saying, one of the things is that there could be differences. There could be important or subtle differences. We're not entirely sure for the different genetic variations. And so the more of you we have in, and to a certain extent, the more of you we also have blood samples from, the more the scientists can help us understand whether the things are the same or different across the group. So I'm just going to go relatively quickly. The scientists know about this. These are cell lines that are available. And for any of the scientists listening to this, we're happy to point you to where you can find them. But importantly, if you go to Safari Base, then you can be able to put in a request to get all of these close to free of charge just for the shipping of the samples themselves. Okay, so let me start out with thinking about what is, I think, the good news in terms of this. So overall, the news is, I think, not going to jolt you from what you've already been experiencing. So I want to just sort of put that out there. We are not seeing major health issues coming up that we didn't expect. We're not seeing major degeneration or things like Alzheimer's, but there are limitations for sure in terms of what I have to tell you. So I'm going to underscore these. Okay, within all of these, I'm going to tell you how many people contributed to what I'm telling you today, because there are certain limitations if we don't have very many people. So for this first measure, this is based on 23 people. And so it's 23 experiences. If you're not represented here, then we may not have represented your experience. Within this, I want to point out, and this will be reinforced with what I go through later, most individuals are in good health. So when we think about this from a medical point of view, how your heart's doing, how your lungs are doing, things like that, people are generally in good health. And we've been seeing this throughout the life course. The majority of individuals are doing fine with sleep, but not everyone. And for those individuals, for the third of individuals where sleep is not going well, I know that sleep sometimes really doesn't go well. Okay. And so within this, there's heterogeneity. There are differences between individuals. I personally think some of those differences are due to differences in those genetic variants. I think there are differences because of that fundamental, even though it's the same gene, different genetic variants in the gene that are affecting the way the gene works potentially differently. And so within this, there are differences, and it's important to recognize those differences. And again, as the numbers increase, we'll have more of specific genetic variants where even within OCNDS, we may have sort of subchapters of individuals with the same variant, and we may be comparing, helping you all to compare yourself within that subtype. So it is, I think, a priority because sleep to me is fundamental, that when you get a good night's sleep, it helps in terms of behaviors the next day. It helps in terms of learning the next day. It helps in terms of all of us as parents functioning the next day. Like there's so many things that this is an important fundamental thing that watch for it. We'll be coming back to in terms of sleep issues. I do think we've identified this as one of the priority issues. Within this, as I said, although most people are alert and aware, there are certain times either with distractibility or because of a good night's sleep, we can see certain issues coming up. In general, in terms, I'm jumping over to the right side here. In general, folks are joyful. They're happy, they're full of joy, they have pleasure, they are able to really experience and exude that warmth and that joy. But I will also say we see about a quarter of individuals that are having not so much joy, right? And so in terms of figuring out either when in the life course is it something else that's going on, we want to make sure that we can support individuals so that we can get closer to 100% for these individuals. In terms of activities, there are lots of types of different activities that folks enjoy doing. With COVID, I think more of the activities have been outdoors sometimes as we've been doing these. But I will say consistency is important, right? Consistency, being able to have expectations, not deviating too much, no curveballs within this. And that's what we've been hearing from folks as we go forward. Within this, in terms of social dimensions, many individuals do like interacting with other people. Not everyone, but many people do. And you'll see it when you get hugs in the hall out here. But I do think there are differences by different time periods. And adolescents, I'm going to show you in just a second, but adolescents or people as they're getting to be young adults especially, some of those things are changing. And so it's important for us to understand those. I am excited about things on the right over here in terms of daily life. For the most part, people are able to continue to improve in their functioning of daily life. And in terms of technology, I'll emphasize this, many individuals really like technology. In fact, I was in some cases obsessed with technology, like really like it. On the other hand, I do think the generation that's coming up has lots of opportunities to be able to use this technology, right? And so for any of you guys who happen to be app developers or programmers or have other friends who are, I think we've got lots of opportunities to be able to use this to expand functionality. Okay, I'm going to focus now in terms of medical issues. And as I said, I would always tell you the numbers. So this is based on 44 participants that were able to confirm that have the right diagnosis. And that's what we mean, as Isabel was saying, in terms of these particular types of variants. So it's based on 44 individuals. Here's a limitation, and I do want to underscore this. This is an age distribution. So on the x-axis here is shown the age of the individual, and this is the frequency or how many individuals participated in the study that I'm going to show you data on, okay? And so when you look at this, the majority of individuals are young, still under the age of 10, and most of them pre-pubertal, so before puberty's coming in. Not to say exclusively, so we do have a few folks, very small number, who are adults, and we do have a few folks who are in puberty, but puberty is a point where things change quite a bit. So I'm going to underscore that. I think it's a gap that we need to fill in terms of understanding better what's happening over different life stages. As folks have been saying, when we look at the average age, we're about 9 years old, and we have individuals, and this actually gives me great hope. We have more individuals who are itty bitty as they're being diagnosed. And so with this, I think those are opportunities to onboard young people and to be able to help share with each other, right, what we've learned from the 10-year-olds, giving that down to the 1-year-olds when they're diagnosed. And then also, as I said, being able to, one of my goals, if anyone has suggestions, how do we find more of the adults who are out here? Because some of those adults, I think, are going to help all of us in terms of what to look for in the future. Okay, all of these are shown in the same way. So let me help orient you to these. The number first here is the number of individuals, and this is out of a total of 44, remember. And then over here in parentheses, we put the percentage. So the number of individuals who had these issues. Not surprisingly, essentially everyone's having trouble in terms of neurodevelopment, right? I would argue that although our numbers aren't quite 100%, it's really pretty close to 100% in terms of individuals who are having issues with development. And I would say most of our issues are what I call above the shoulders. In other words, most of the issues are what we think of in terms of the brain and behavior. Within this, we do have, not surprisingly, a significant proportion of our community that has autism, and then also anxiety and problems with attention. And within this, I will say that sometimes this needs medication, needs treatment. It may need ABA therapy, may need educational strategies as well. But in certain cases, it will need medication. And as we've seen the children get older, that's when we've seen more and more use of the medication. I won't say that we have one single medication that's like the cure-all in terms of this. And so right now we're still doing a little bit of trial and error. And my hope is that over time, there's less of the trial and error and there's more of getting to the right medication by sharing our information together. But this is where we're seeing the largest part of the issues. Okay, one issue that's come up very specifically is in terms of toilet training. And I know some of the groups have talked about this already, so you may have been one of those breakout sessions yesterday. The good news here is that in terms of being able, if you have really little kids that haven't yet made it to this milestone, the good news is I would say there's hope in terms of being able to achieve the milestone. I'm not saying it's trivial. I'm not saying it's going to happen overnight, but the majority of our young people in a reasonable period of time are getting to the point where they're dry. And so in terms of being able to be in more activities, being in more sort of preschool programs or other programs, the good news is they're getting there. With this, it's coming a little bit later in terms of bowel movements. So number 2 in terms of things, but it is coming. And so again, being patient in terms of thinking about doing this, working with behaviorists, hearing about some of the things from Nate yesterday, I do want to be encouraging that this is getting there. I will say sometimes there's a step when you take a step back. So with this, I don't think it's a permanent step back all the time, but there are times when, you know, there may be a little bit of regression for something that's going on, something that changes the order of things. And so, but don't give up in terms of continuing to try and move forward and getting back on track. Okay. In terms of other things that we see that your neurologist may have said, and these are terms that they use, so I'm putting those in the same terms. We see individuals that oftentimes have problems with low muscle tone, so their core is low, and that's why you're working with the physical therapist also. That also can one other manifestation that we see are problems with coordination. So kids tend to be not so coordinated. They're bumping into things, they're falling more. And it's a combination of these two things with low muscle tone and coordination issue. It does get better though with your physical therapist. So in terms of doing that, it's repetition. I know it feels like it takes forever. Definitely extended school year is helpful in terms of around the year so that you don't lose those skills as they come up, but definitely working towards those. Those are the main issues that we see, and movement disorder, I should have said, tied in with that, the main things that we're seeing. Don't worry so much about things like whether the head size is smaller or larger. It's not going to explode. It's not like there's going to be hydrocephalus. It's not like shunts are needed. It's not like things are shrinking, but those are things that we've seen. And again, I hope at some point we'll be able to get to growth charts so we can actually help pediatricians know for OCNDS, here's what you should expect. Don't freak out about this. This is just normal growth. This is normal head size. So that people aren't so concerned about some of these things that come up. Okay. Beyond that, and this is an important one, seizure disorder or problems with epilepsy is something that a significant fraction have. So about a quarter of the individuals in our community are having problems with epilepsy or seizures. Within this, I want to say and just highlight here that the average age of first seizure was about 3 years old, but there's quite a range that we've seen in the community here. So anywhere from itty bitty all the way up to teenage years. And so if you haven't, in other words, if your child is still 8, although it's likely that they've are less likely to have seizures, because again, the majority of people don't, you're not quite off the hook yet in terms of when we've seen it in the community. So with this, the reason I emphasize it is because seizures are treatable. And so making sure that if there's a movement, if there's something you're seeing, you're not sure of, bring it to the attention of your pediatrician, or if you have a neurologist, if you're able to videotape it. And I know in the heat of the moment, it's not the first thing you're thinking about, but if you do have someone someone that takes a videotape of that and shares it, it's oftentimes helpful in terms of us knowing whether or not it really is a seizure. Within this, there are various different seizure types. I'm not going to worry about it so much for you guys. I put it up here that if they're neurologists or providers out there looking at this, they have the information available to them. But for a significant portion of our community that have had seizures, they are continuing to take medication for the seizures, right? In some cases, it's not because they're active seizures all the time, but their neurologists are trying to make sure that the seizures are at bay and making sure that therefore they're going to be safe and also be able to think more clearly and learn better. And so important to recognize these and get on the right treatment if it's necessary. Within this, the news otherwise is good. So I just want to, if it's taking a time to let this sink in, just to say the news is good. We have had some individuals with congenital heart disease. If it's there, it's present at birth. So if you've had your child's had an echocardiogram, they say there's nothing there. You don't need to worry that all of a sudden this is going to pop up. It's yes, no, it's there, it's not there. And in general, very few individuals who have needed any treatment or surgery for this. So this is good news as we think about this. Surgeries in general, there have been a few individuals, and I know it may be hard to see, but these are small single digits, 5 to 7% of individuals who have needed surgery. A large part of it's been tonsillectomies or ear tubes, so minor kind of kid surgery that we see. We have had a few folks who have had problems initially in terms of feeding and needed gastrostomy tubes. And so that's been helpful in terms of getting them stronger, getting their nutrition, especially when they're little. And many of them can outgrow that and have those gastrostomy tubes taken out over time. And again, low numbers here. We have had a few of the boys who've had undescended testicles, and within that, it's something your pediatrician can check for simple surgery to tack those down within the scrotum to prevent later cancer, later testicular cancer. But again, not very often, and when it's there, very treatable. And so not a big issue medically. The one issue we've seen with fair frequency is issues with growth. And so this is why I said I think it would be great if we had an OCNDS specific growth chart. Our folks do tend to be shorter. They do tend to be, whether it's called not gaining weight so well or not growing so well. This is an inherent problem, or not problem, but an inherent characteristic of the condition. And so again, I don't necessarily want your kids being pumped up with lots of calories just so they grow this way. We want them to grow this way, right, in terms of this. And things like people often ask, well, is growth hormone going to help? We haven't been seeing that the children are deficient in growth hormone, so just giving them shots of extra growth hormone are not going to suddenly normalize their growth and make them be full size. So as you're doing that, again, lots for the doctors who may be watching this. One other issue I do want to emphasize are vision issues. And the reason I emphasize this is because they're treatable. And if you're not seeing clearly, if things are fuzzy, or if you're not able to focus or your depth perception is off, it just makes it that much harder to coordinate your movements, to be safe, to learn, to take in the world. And so if your child hasn't seen, especially a pediatric ophthalmologist or an ophthalmologist, especially for new folks out there, I want to make sure they do. Many of these things are treated either with glasses, patching, or simple surgeries. And so with this, you as a parent, if you're seeing a lazy eye or you're not seeing both eyes focusing forward, easy thing to then get a referral from your pediatrician, see the ophthalmologist. It's best if it is specifically a pediatric ophthalmologist who sees children with special needs, because it's oftentimes hard to other guys get someone to cooperate with the exam and to see what they're really seeing. And so again, oftentimes big children's hospitals will have the right eye doctor that you need to see. Within this and putting everything together, the one thing I haven't mentioned so much are the tummy troubles. So gastrointestinal issues are also something that we often see, combination of either diarrhea, constipation, or even both, depending on the day. We don't have great sort of answers in terms of this, except to say that commonly we're seeing folks using medications for reflux, medications for constipation, and combination of diet. And the more that the kids are active, the more things it keeps things moving through their system. And this also relates to the potty training we were talking about, or toileting. So all of these things kind of fit together as we see this. Otherwise, what I take for this is that I know it's hard to see, but these numbers are low. So the percentages are low. We're not having catastrophic issues coming up, with the limitation that I don't feel like we've got great representation from adults yet. And so there's still more to be learned in terms of what we need to do to take care of our folks as they're getting older. I did put here in terms of just so you could see some idea of how serious conditions were by whether or not people were using medications. So I took that as being a sign to us that they were serious enough that they needed to take medications. So I started talking about this earlier, but the tummy troubles, we're seeing a little over a third of individuals that are taking medication, largely for constipation or diarrhea. As I said, about a quarter of individuals for epilepsy and about a quarter of individuals for sleep. And so sleep is, for those individuals that are having the issue, it is significant enough that they're taking something for that. Oftentimes it starts out as melatonin for the sleep, something that in the United States, at least you can get over the counter, but increasingly folks that are having significant issues needing to go up to stronger medications. Otherwise, most of the other things that we're seeing here, things like treatment for asthma are pretty common kid sort of things, not things that are specific for OCNDS. But I will point out that things like hyperactivity, whether medications, I won't say there's one single winner in terms of medication for that, but we are seeing that it improves and that ability to focus in school is improving, the ability to learn better is improving. Okay. With this, the numbers are ridiculously small. So I actually even wondered whether we should include this, but to say that phenobarbital, which is a very easy, well-tolerated medication for epilepsy, is working and a best medication for some people. And so I don't think it's necessarily requiring multiple medications for epilepsy or some of the really advanced or expensive medications. It's kind of standard things for epilepsy oftentimes work. So the neurologist out there, that's mostly for you guys. Okay, so this is now something that we look at for development and to see how development changes over time. So again, I told you I would do this. This is based on 17 individuals. This is age in years along the x-axis here. And this is a scale of how they're developing. And this is showing their developmental age in years. So if you were taking a typical child, this would be, hopefully, if you were age 5, you would be acting like you're age 5. Now, in this case, that's not the case, right? So this curve that should look like this instead in our kids is sort of bent down here because they're developing more slowly. On the other hand, they are developing, and I want to emphasize that, right? They are making forward progress. It's not like they're going like this, where they're going downhill. Okay, each one of these dots is a different person. So one of the things that you can see is that we have lots of folks that tend to be sort of, and this line here is an average of all of those people together. But the point is that each dot is a different person and there are differences between people. So we've got someone up here that's doing very differently than others. We've got someone down here that's also doing very differently than others. And so the point that I was making before is that different genetic variations may act differently. There may be some differences person to person, genetic variant to genetic variant. And so it would be ideal if we can get the numbers to be large enough that we can look at each variant and sort of separate them out and seeing if we can use that to help you better in terms of expectations as well as which things are working. To give you a sense of what this means, because these scores mean nothing, right? In terms, they don't mean that much. This is just showing you, Dr. Green Schneider was showing us here that this is showing us being able to use single words. And so being able to get above this threshold means all these folks are now able to use single words. We're not quite there for everyone yet, but we are getting there for most people over time. Showing a different dimension of this is thinking about being able to take care of yourself. So around what you need to do on a day-to-day basis, things like being able to feed with a fork and a spoon, being able to dress yourself or undress yourself is even easier. But again, showing that folks are making progress over time. Some folks are making really good progress over time. So I want to put that out as a goal in terms of being able to achieve that. And I think a lot of us are focused on very practical skills in terms of what we can achieve over time. This is thinking about sociability, so interacting with other people, appropriate behaviors, being able to make friends or have friendships or relate to others. And this one in particular, our slope you may have noticed is not quite as strong. So in terms of how much progress, we are making progress over time, but we also have a lot of folks who are below the curve here, right? And so this is related in part to the autism or autistic-like phenotype, or rather behaviors that I was talking about. So whether it's things like being able to show interest in friends or being able to make eye contact with folks, we have a lot more challenges here. And I think that's something that many of us are working on in terms of with our therapist. And then in terms of being able to do things like hop, skip, jump, go up and down stairs, again, being able to make improvements over time. And I love watching the kids here in terms of safely going up and down the stairs here, but I'm just thrilled in terms of being able to see how many of them are able to achieve those milestones. Okay. So the other thing is that we have measures to be able to see, are they having behavioral problems that are pretty significant? And so within this, I'm going to divide this up into our younger folks. So these are folks that are age 1.5 years to 5, and we have 20 individuals who are in this age range. And again, so these are itty bitty ones. Now, the good news is when they're little itty bitty like this, we're not having big problems in terms of major diagnoses. And so that would be in this range here where we see significant concerns. And you'll see that none of these behaviors, whether it's aggression, sadness, anxiety, attention problems, oppositional behavior, no one's in the significant concern range or even in the mild range. Okay, so that's good. But this is the first stage of life. This is when they're still in preschool. Next, we're getting up to folks, and we have 26 individuals in this range who are now age 6 to 18. And I will say that's a pretty wide spectrum for me because within puberty, things are changing quite a bit. Now, the good news is within this, again, if we look at same categories we were talking about before, and now if we look at significant concerns again, the good news is we're not seeing individuals with significant concerns or even mostly with mild concerns. But I will warn you that this is a group average. So these are individuals across the whole group, and there are specific individuals that are buried within here that I know are having issues. So I don't want to say that, you know, everything's bright and rosy and no one's having any issues whatsoever. There are certain individuals, especially as we're getting into adolescence, where the particular behaviors can become, I will say, overwhelming. And so one of the things we're trying to do is have more individuals who can help us understand what those issues are. And in particular, we've been sort of hovering and looking very, very closely at this era of the preteens and the teens. So age 10 and above, as our group is getting a bit older, and as we can try and prognosticate what the issues are going to be, and specifically either medications or behavioral strategies or educational strategies, because I know they can be extremely stressful for those families. Okay, so as we're doing this, just in brief summary for the young ones up until age 5, we're seeing some issues in terms of just impatience. They want what they want when they want them, and they need to have it, right? And so I'm just watching the nods, and some of you are very much there. So, and with this, you know, things are, you know, they need to be the same, or the more same they are, the more you can sort of not have curveballs for them, the easier the day is. For our older folks, as they're getting into the teenage years, we're seeing more issues in terms of obsessions, sort of a little bit of doing the same thing over and over, whether it's picking at something or whether it's a particular more ritualistic behavior. Problems with attention and distractibility are coming up a lot. And then sometimes in terms of irritability. And I think this also, as I said, has somewhat to do in terms of the sleep issues that are coming up. So overall, good news is we don't have so much in terms of medical issues, with the exceptions of seizures. Watch for seizures, important to do, and eye issues. So making sure that those two things are the important things to keep an eye on. Otherwise, in terms of other things and growth, I will say it's not, you know, terrible problems, but don't be afraid. Don't be totally concerned if your child is not growing, is not quite as tall. Otherwise, it's mostly about behavior, education, and being able to figure out the strategies that are going to work best. And that's, I think, the next chapter in terms of where we're going to focus a lot of attention. Okay, let me just also say, and for any of the researchers in the room, feel free to correct me or add to this. I was so incredibly excited about the day that we had. It started out on Thursday, it rolled into Friday, it's continuing on to Saturday, but you have an incredible group of dedicated researchers here around the world who have been at this for many years now. They started out pre-COVID. I can attest to throughout COVID, they were on Zoom, working with each other, strategizing, figuring things out, and seeing them here together today and throughout this weekend has just been fabulous. Let me take at a very high level in terms of what they've been able to do. They've been able to do very detailed work from looking at the structure and the protein and how it works in terms of it has this function as a kinase. So it actually, I think of it kind of as like a conductor for an orchestra, and it's being able to regulate the amounts and how different proteins work, how the flutes, how the clarinets, how the bassoon, how the tuba, how the percussions and the bass are working, and making them work at just the right level, just the right volume, and to coordinate. And that lack of coordination and having just the right volume of those different instruments is part of what causes the problem. But we're still at the stage where we're not quite sure, is this mostly a tuba problem or is it mostly a clarinet problem? Or maybe it's a lot of both of them. And so they're doing a lot of work in terms of trying to figure all of that out. And I was joking with them earlier this week. I said, it's a zoo out there. And it's a zoo because they've been actually making model organisms. So things that we don't have to experiment on your children, on your family members, they've been making critters. So some of you saw the reptiles last night. So they've been making critters that go from zebrafish to flies to mice to frogs, all of these different things so that they can be able to study your particular family member's genetic variation and being able to study it in a living, breathing organism to try different treatments, see if different treatments. And when I say treatments, I'm talking about things like medicines that might help to be able to make things better. So they've had a lot of progress. They've made a lot of traction and a lot of people. I can't, I almost feel like, sometimes we have a joke that we have as many scientists as patients working on these conditions. I think we're just about at that level because you've got such a huge number of scientists out there working behind you. And so they are making a lot of progress, and I know many of you have, but do thank them for what they're doing, because it's been, I know under COVID especially, it's been really hard for them to keep things going, and they've really doubled their efforts in terms of doing that for you. So I'll stop here and I'll be glad to take questions either from those in the room or anyone who is at home watching. Feel free to put your question in online.
We're going to start for the final portion of our gene morning. And we're going to finish with our last presenter, who's Dr. Cora Taylor. And you may recognize her for many of the Q&A sessions that they do at Simon's Searchlight. She also will tell you that she's a principal investigator, and her CV is very, very long. So I'll let her do that. But she's going to give a short presentation, and then you can also ask her any questions that you have about strategies for sleep, toiling, behavior, early intervention, education, sensory issues. After she's finished presenting, we're going to have all of our presenters come up, and we're going to do the Q&A then. So hopefully you've had a little bit of time to digest all the information that you've received today, and any of the questions that you have, we can ask during that time. Just a short housekeeping note: at 12:45, we're going to take a picture right here on the second floor. And now our beloved Cora Taylor.
Thanks very much. I'm really happy to be here and to meet all of you in person and be here in person. I'm just going to stand up front because it's a little more comfortable for me. And so as Jennifer introduced, I am a psychologist, and I work with Simon's Searchlight as part of the team that's based at Geisinger. We do a lot of the recruitment and retention. If you had your blood draws, those are all my people. So we're really happy to meet you guys in person. Again, I'm really happy to answer any of your questions about behavior. And I have a preloaded question that I got from Jennifer kind of in advance about some of the issues that you guys have shared about things that you've been struggling with. So I will start with that. I do think of this as being a kind of very informal kind of format, really and it is an ask me anything. And if I can't answer it, we have some people in the back who will write it down, because there might be other people here who can, or we can try to figure out who can answer that. But some of your more specific questions about medical issues, medications, those things would be out of the scope of what I'm able to answer based on my training and my licensure, but anything more related to behavior. So I'll start out a little bit with the preloaded question, which is that how many of you have an individual with OCNDS in your family who struggles with things related to sensory? Okay, that's about what I was expecting, is that sensory things are really, really common. So I just wanted to take a minute and sort of talk about how we think about sensory issues and what those things mean, and also the fact that you might hear a lot of conflicting information. So a lot of times what I hear from families is, does my child have a sensory processing disorder? Is that a separate diagnosis? Is it in addition to OCNDS or other things that you're struggling with? The difficulty with this idea of sensory processing disorder is that it's not a diagnosis that's actually in what we call the Diagnostic and Statistical Manual, which is this big book that psychologists and psychiatrists use to diagnose different conditions. So it hasn't been recognized broadly as being a separate disorder that would be something in addition to what you might get. That's not to say that there aren't some individuals out there, especially you think about occupational therapists or others who really focus on sensory issues, that might assign that diagnosis, but it's not something that the medical community will necessarily recognize as an official diagnosis. That's not to say that your children are not struggling with sensory issues. We know that having sensory sensitivities, hypersensitivities, hyposensitivities are definitely a part of autism spectrum disorders, many children with ADHD, intellectual disability, which we know, and learning disabilities. We know a lot of children with OCNDS struggle with those diagnoses, which are official medical diagnoses, but sensory issues are a part of all of that. Sometimes we'll see both overreactivity to sensory issues. So you might see that, your kids maybe are covering their ears for sounds. Sometimes sounds like you don't even know, like, oh my gosh, I didn't think that was that loud, or it might be a really specific sound. You might see that the kids are covering ears. And then at other times, they're fine with things being really, really loud, or they're seeking out things. That would be sensory seeking. And often we'll see that children present with kind of mixed profiles in that. So there might be some sensory things that they're seeking out, and then other sensory things that they're avoiding. The other thing I heard from Jennifer that a lot of you were talking about is this like fight or flight or freeze, kind of that anxiety reaction. And sensory issues are often very much tied into that. So how do we respond to sensory sensitivities when we see them? So I just wanted to start out with that little kind of introduction of like sensory issues are something that need to be addressed and are important and are part of what's going on in OCNDS based on what we're talking about. You might be hearing a lot of conflicting information from your providers about what that actually means. But I'm happy to answer any specific questions that you might have about the sensory issues that your children are actually experiencing. But just wanted to start out with that little brief introduction. So if anybody has questions, there's a mic up here, or I'm happy to go to you. Yes.
Can you describe what are sensory issues and how they sometimes come? What do they look like? What can they look like?
Yeah, so you can think about sensory, like if you think sensory issues, you have 5 senses. And so sensory sensitivities can present, or sensory issues, and think about issues. So you can have a sensitivity or you can be sensory seeking. So it can kind of go either direction. And like I mentioned before, you can have a mixed presentation where your child might be sensory seeking for some things, but sensory avoidant for other things. So if you think about touch, that would be one of your senses. So you might have kids who really are seeking, sometimes we think about kids that are like running into walls. They might be banging their head. Sometimes it's a soothing thing as well. So you might see that some children are banging their head in order to help themselves fall asleep, or they're rocking a little bit to help themselves fall asleep at night, or there might be some sensory sensitivities in the touch realm. So tags in the backs of shirts might be a way that presents, or certain fabrics that your children don't like. Another sensory seeking for touch might be, you might see that your children are rubbing things on their face or touching things to their chin, those sorts of things. So they're seeking out certain sensory things in that realm. So you also have visual sensory. So sometimes you might see that your children are either oversensitive or undersensitive to things like lights. We also sometimes see our kids like really want to look at things and inspect things, or maybe look at things from the side of their eye and do some peering. So that would be, we would consider that to be a sensory difference, a sensory issue, sensory seeking. Noise is something that we hear pretty commonly from families who have children with developmental differences. And so we might see that children are covering their ears for certain sounds. Sometimes it's not necessarily even that the sound is loud. It just is a different way that's being processed through the brain. We do all have, even within this room, even those of us without OCNDS will process sensory things a little bit differently. It's whether, I think the key word there is issue, whether or not it's an issue. Does it interfere with your ability to interact with others, to access school, to be a part of the community? That would be when I would consider it to be an issue. So we all have differences. Is it an issue? So that's when I think when it is an issue, that's when you need to think about, do you need to address it? So is it something that warrants intervention, that we need to target that concern? Because from my perspective, if that sensory issue if it's not just a sensory difference, if it's a sensory issue in that it's interfering with your child's ability to access their therapies, to access their learning environment, that's when we maybe need to really think about, okay, how can we help this child, how can we help this child move forward in a way that they can continue to access their community and access their learning? Does that answer your question, Jennifer?
Yes, I was also wondering who would help to diagnose whether or not, or observe your child to figure out whether or not they're experiencing sensory issues that are interfering with their ability to do different activities, whether that's school, sleep, go out on an outing with a family. toilet, those kinds of things?
So I think the primary person would be whoever is intervening in that realm. So I mean, I think it's multidisciplinary. So it's not necessarily one person who's seeing it all the time. And if your child is experiencing sensory issues in one environment, but not another environment, that might be an important part of the question. So what's going on in this one environment where this seems to be an issue, but if you experience the exact same sensory thing in another environment, there might be something more complicated going on than just saying that this is a sensory difference or sensory issue that's going on. So I think it does need to be multidisciplinary. So I would talk to, if you have a child who's struggling, say in school, I would talk to everybody on that child's team and see when is this happening? How is it happening? So what I'm essentially describing is what we call a functional behavior analysis, or an FBA. And the purpose of an FBA is to identify why behaviors happen. So we think about that being kind of 4 major categories for why behaviors happen. So I either want to access something that I like, or I want to get away from something that I don't like. And that like sensory can be a piece of that, but it also could be other things going on, other things potentially going on. So I would think about, if it's really interfering, I would think about doing a functional behavior analysis. The person who does that is the behavior specialist on your team. You might hear them called your BCBA. If you don't have one on your team, you can ask for one, or try to find somebody in the local community who can provide that service. Many school systems do have somebody on their team who is capable of doing an FBA, even if it's not your child's behavior specialist. It might be the school psychologist who's capable of doing an FBA. So I think that's the first step, to make sure that what you're dealing with is really truly a sensory issue, is to start with that behavior analysis. So why is this behavior happening? Because sometimes, and also it could be a multitude of things. So sometimes when a behavior might start because it's sensory, but it might continue for a different reason. So it might be that, I didn't like this crowded environment. And so I got really upset, and I got to get out of, say, going to that field trip or going into that major, special school day that I didn't really want to go to. But now I know that if I get really upset when it happens, I get to get out of it right away. So sometimes it can like perpetuate as well. So I think there's a lot of issues going on. I think a multidisciplinary approach and talking to everybody on your child's team, figuring out when these behaviors happen and how to handle them is the right next step.
And when you're not of school age, so it's before you even enter school, or you've graduated out of school, and you might be seeing kind of potentially something that you could think was sensory, or you want to have it evaluated, how would you go about doing that?
So if you're not school age yet, but you have early intervention involved, I would start with them and see if either they have somebody on their team who can help bring somebody in who knows how to do that. The BCBAs also do have a website. I think it's a little counterintuitive for behavior specialists. It's BACB is their actual website, where you can go online and find a BCBA who might be in your local community who could help you. And they might be contracted with different organizations. And then you could go to that organization and try to bring that person in, but it's at different ages.
And I know in this group, I'll just continue on, that we hear a lot about kind of weighted blankets or different tools to deal with sensory issues. Assuming, because in many of these instances, we assume people have robust teams or even cooperative teams, and we don't necessarily always see that. And so what are some of the things that parents would want to think about to start throwing out ideas? I even know in our own group that someone went to Walmart yesterday, right, to get a weighted blanket to start to try that for sleep. Anyway, with that being said.
Yeah, so I will weigh in on the weighted blanket. One very quickly is that if anybody has an infant with OCNDS in the room, we do not use weighted blankets. They can be very dangerous for younger kids who you need to weigh enough in order to use a weighted blanket. So keep in mind that you have an infant, younger kid, weighted blankets, no. The second thing is that most of the evidence that we have for these is anecdotal. So, parents come into my office, they tell me, should I try a weighted blanket? And if I go to the research literature and try to find a really robust, well-designed study that says, do weighted blankets help or not? It's not there. So a lot of it is trying it. And if it's helpful, And it's, you know, if, as long as it, so my true north is if it's not harmful to your child and you feel like it might be helping, I'm okay with it. But as long as you're still investing time and energy and at times money into things that we do know work. So we do know that some of the behavioral interventions work. We do know that getting speech therapy, going to outpatient, we do know that trying augmented and alternative communication devices to help our children communicate. We know that those strategies really work. And if there are other things that you might want to try that we know are not harmful, then if you want to try it, I think that's okay, as long as you're able to still adequately evaluate everything else that you're trying. So if you're really struggling and you go to your doctor and you want to try a new medication, but you try that new medication, but you also go buy a weighted blanket, you start doing other things that we don't really know if they work, then we can't evaluate which one helped. So I think as long as you try to think about it, like if I was designing an experiment to figure out what's working for my child, I would start one thing at a time. I would try it for a while, for an extended amount of time. We want to try things for longer than just a day. And then I would ask people who didn't know. So I would say, hey, you know, like maybe to the babysitter, or hey, maybe to the teacher or the speech therapist, hey, have you noticed anything different, or how's he doing? Is he doing anything? And then we would get information from multiple sources to try to identify if things are actually working. And I think that is the major limitation of some of the research, is that some of these things we really just don't know. The evidence that we have is from hearing from families that, hey, this seemed to work for my child. Yeah, so the question is, a child who, when somebody is talking close to them, that there's maybe get a tingly or a tickly sort of feeling. Yeah, that specific situation is not something that I've personally heard. It could be sensory, it could be anxiety, right? There could be a lot of things going on that might create, if you think about like what makes you sort of like, sometimes you can feel tingly if you're anxious, and it's sort of like how your body is interpreting. How old, may I ask? 11. So you think about too, so when kids are developing, so I have a question for you back. How do you know when you're feeling anxious? Yeah, can you describe that feeling? Like how do you feel in your body? Your heart beats, yeah. Yeah. How do you feel when you're happy? What does that feel like? Yeah. So for like me, if I feel happy, I might feel like a little jittery, or like if I'm excited, I might feel a little jittery. I might get like, we would call butterflies. Like if I'm scared, I like have an internal feeling of that too. So when kids are growing and developing, when they're really, really young, they get those feelings, but they can't interpret them. So they start to understand. So when kids get to be about developmentally 8 years old, that's when we have what they call metacognition. So I can understand like how my body feels. And I know that means I'm getting anxious or I'm getting scared. And that's the reason that I say 8 developmentally, is that that's when some of the like evidence-based therapies for anxiety start to work, because you can think about how you're feeling. You can start thinking about your own thoughts. Prior to that age, you're having a lot of feelings, but the children aren't always able to like catch themselves and interpret that and actually put that into like how my mood is. And that's why before about age 8, some of these cognitive behavioral therapies or like therapy, one-on-one talk therapy is less effective, because we can't teach, we have to help children learn, okay, if you're feeling that way, this is the way that you're feeling. So I haven't heard that specifically, but it could be related to sort of a mood or an emotion that she hasn't figured out how to interpret yet. Yeah. Mm-hmm. Mm-hmm. Yeah, no, she's not able to label her emotions yet. Yeah. Questions? Yeah.
How do you help support a sensory seeker when they're presenting things towards their mouth and the chin in more of a safe way? So we've had issues where my daughter has swallowed things because she brought them close to the chin of her mouth, and then they landed in her mouth and then she swallows them. So how do we help support that sensory seeking behavior in a safe way, but still allowing her to kind of get that touch and that feel.
Yeah, great question. So the question for those online is, how do we help a child who is sensory seeking that might be putting her in danger, like touching things to her chin or her mouth and maybe accidentally swallowing them, do that in a safe way? Have you looked into getting her like a necklace at all, like a chewy necklace? Okay. Yeah, for many children who are having sensory issues where they're putting things into their mouth, that's often what we'll think about doing. So what we call that in more formal terms, we call that a replacement behavior. So what you want to think about if your child is engaging in things that are maybe unsafe or dangerous, and you want to think about how can I help support them through this, is you want to think about, okay, how can I let them still engage in that behavior, but in a way that is safe? So a replacement. So thinking about having something that's accessible to her, so she could put that to her chin or in her mouth, but it is something that you can easily wash, you can replace. You should probably get a couple of them, so you could give her a different one, and then you could like wash the one that she has. And so that would be the first thing that I would probably try, is to get her something that you would feel safe with her having with her all the time, that's, you know, physically actually near her. So if she tends to grab things that are like lower, you could think like play with the length of the chain too, based on what works. So sometimes the chewies are like a little here, so they're really close to the mouth, but that's not where her hands are. Her hands might actually be a little bit lower. So you could think about having it hang a little bit lower, so it's easier for her to pick up and access to put to her mouth. Yeah. I don't mind repeating the question.
And I tried all of these I think
Yeah, so we've seen some other families, not necessarily with OCNDS, but who do seem to have some repetitive hand biting. And the first thing I would do, my first thought is, how do we keep him safe? And the first thing I would do is think about having some sort of a glove that you can put on him so that when he bites himself, he's not hurting himself. And because there are some compulsions that are like things like that, where it's sensory seeking and it's very repetitive and it's very, very difficult to get under control. And so the first thing I would do is, okay, how can we keep him safe? Can we put something on his hand so that when he bites himself, he's not actually hurting himself? And then you can think about, keeping his hands occupied. Is there something like, would he have a fidget toy that he would play with, so that we're keeping his hands constantly occupied so that it's not coming to his mouth? Those behaviors are very, very difficult to get under control without using what we call punishment procedures. And we typically don't recommend doing that. They're pretty aversive. They can take a while. So we had a kid who was engaging in some doing this, like he was chomping his chin. And it was a struggle. And the only way to get him to stop was every time that he chomped his chin, we had him dump out a bucket of blocks and put him back in. And it eventually got it under control, but it's aversive. It takes a really long time. It's sort of like a last resort. So if we can keep him safe, potentially that would be the thinking about just having a way to keep him safe while he does that, because it is really, really hard for some of those very sensory seeking behaviors. Yes.
So I goes off what they were saying with COVID. My daughter chews on her medical mask, like any mask that she has, she chews full in them through school. They pick them up, they like soaking where, of course, can't use them after she's full in them, and she doesn't, she's like the jewelry or any pieces. We tried to be like, no, explain to her, those sorts of things. How can we get them to stop?
So you've tried, but she hasn't taken to doing anything else.
Yeah, like we tried to can't do it, or try this instead of that, or she doesn't chew on shirts or anything else. It's just some mask around her face, something instinctively. I don't think that's something we should.
Yeah, well, it's hard too, because once you have the mask on, you can't access anything else really easily, right? So you have your mask on, and then if you had a chewy or anything like that, it's not just like you can bring it to your mouth. You'd have to take your mask off and then bring it to your mouth. So when a child is having trouble kind of learning the replacement behavior, what you'd like them to do instead of what they're currently doing, oftentimes it does take a fair amount of teaching to get them to that point where they're understanding what you would prefer that they do, or what might be a safer choice for them to do. So the first thing I would do, so every time you're going to have to get her teachers involved, because that's where most of this is probably happening, because you don't make her wear a mask at home. So the ability for you to teach her to do this is limited, and it ends up kind of being on her teachers to make sure that they're consistently implementing the intervention. Now, you could have her wear a mask at home and try to help teach her, but if we're getting to a point where hopefully we're not wearing masks, and nobody wants to wear a mask in their house, right? So I think you'd really have to get the teachers on board to help teach her that when she does this, every time they see her do it, they need to go over to her and instruct her what to do. And I would say, like, use very clear, matter-of-fact language. So making sure that we're not getting upset at her for chewing her mask or anything like that, but just saying, no, do this instead, and actually tell her what you want her to do instead of what she's doing. But she might need to be instructed that in order to do this, that there's a couple steps involved. And that also plays into other issues that some of our children have, like, are there fine motor things going on? Can she get her mask off and then actually put it back on? So there's a lot of components to it, but I think, getting the teachers, that you'd have to get the teachers on board to really provide her with direct instruction about what she's supposed to do. Sure.
So how do we differentiate between a sensory need, an anxiety need, or manipulative piece? Because all those components I see my daughter having, where we know that she can do something, whether it's at school or home. With ABA, she tends to do a lot of things for the ABA therapist, but implementing them with me or with him or with my mom is, well, I'm not going to do it. Or it's like a different state. Like it just goes from 0 to 100, where she just.
Yeah, so that's a really good question. And it's really hard because I think anybody in here is a parent. Like, I mean, I can go up here and give you the best advice theoretically in the world. But if I went home to like my niece or my nephew and you had like a little camera on my shoulder and you watched what I actually did, I may not be taking my own advice. So it's really, really hard when your mom and when your dad and when your gram to actually do the kinds of things that they're saying, because it's your kid, and that's okay. But what I will say is that I think talking to your ABA therapist and figuring out as a family, what's the most important thing right now? What kinds of behaviors do we really want to see at home? And maybe you're only picking 1 or 2. Like, these are the behaviors we really want to work on right now. These are the ones that matter. And the other behaviors, you do have to pick your battles. You can't pick every single battle every single day. So if you 3 on a team decide these are the ones, this is what we're going to work on right now, then you've already as a team kind of said, this is the stuff that matters to us as a family. This is what we're going to work on with her right now. And then what that helps you do is when you pick your battle, you have to follow through with it, because that's the biggest thing with behavior intervention and with tantrums, is that when you pick the battle, you have to follow through. If you don't follow through, what ends up happening is that she learns that she tantrums, she can do what she wants, right? And sometimes when you start, if you guys all, have like a little team family meeting and decide, okay, these are the different things that we really want to work on. Here's the kind of battles that we're going to pick. And and then having that support to follow through. And also knowing that sometimes behavior gets worse before it gets better. So whenever I go to a vending machine and I like hit the button for water, I get my water every time. And if I go to one and I hit my button for water and a water doesn't come out, I might hit that button a little harder. I might like smack the machine. Like, you might see people like kicking it, because it's like, this has worked every single time. I always get my water, and this time I didn't get it. And that's the same thing when you're trying to work on behavior change for kids, is that if her strategy was to have a tantrum and she knew that it like, oh, I won't have to do that, it might see that she might escalate a little bit. And you should take that as a sign that what you're doing is working, that she's realizing that she's not getting, like, the things aren't going the way it usually does. And so first thing she's going to do is try harder. And then behavior change happens at that point. Yeah. And I think this is the last one, and then Q&A. Okay.
This is about like repetitive and STEM-like behaviors. So my daughter engages in a flicking of a string in front of her face to kind of get that sensory look. Is this something that should be encouraged or discouraged, maybe provided as a reinforcement when her work is completed, because at some points it does prohibit her from working, more like in an OT setting where she has to string beads on a string. She gets very distracted. It's an item that is with us 24/7 all the time. So is there some kind of
I would never
encouragement?
punish it. I would not necessarily discourage it, but I don't think you need to encourage it either necessarily. So by encourage, I mean like say, yay, keep going, like encourage. It's okay for her to access that, right? That is providing a need for her. And it's, you know, if you think about, all kids have ways to relax, to calm down, to have fun, to do good things. And there's nothing wrong with it. She's not harming anybody. She's okay. And so I think you could potentially use it as a reward if she finds that rewarding. But yeah, in some cases you are going to have to remove it from the situation in order for her to engage. But if she can understand, first we have to do our work and then you can have your string back, that's fine. I'm completely fine with that. Okay. Perfect. Okay, I think we're going to regular Q&A. Okay. Yes.
So as you know, I have a girl who's going through puberty currently. Do you have any recommendations for help with when her menstrual cycle comes, whether it's medicine or specific doctors, books or anything like that?
I'll let Wendy address maybe the medication question. I think in terms of puberty, there is a wonderful online resource at Vanderbilt through Vanderbilt University. It's called Healthy Bodies, a puberty toolkit for girls, and they have one for girls and one for boys that has a lot of really excellent strategies about preparing for puberty, practicing like wearing pads before her period comes. Is she independent with toileting? Okay, so my goal is if you're independent with toileting, my goal is always independent managing periods. And how are we going to go about doing that? So there's lots of ways that you can use other strategies. So thinking about, how do you already teach your child? Do you use visuals? Potentially using visuals might be helpful. How do we know when it's time? How does she know when it's time for her to change her pad? The reason I say pads is pads are a little bit easier to use. Getting the kinds without wings are easier to use. Unpackaging them in advance, that makes it a little bit easier as well. If she has any struggles with fine motor skills, and also just being prepared. So since she's 11, I would actually start preparing now, getting used to, I don't know, does she wear a bra yet? Just started. Yeah, and I think like all those little practicing things, like, so you can start with a training bra. They also make period pad underwear too that you could think about using for her. They are a little bit more expensive than just using pads, but it could be an option where she has period underwear, and we could teach her that when she gets her period, we know that this is what she uses. And also just making sure that she knows now in advance, like when her period comes, she's not hurt and she knows to tell you. And then you can also help her, if she can use a calendar, start tracking so she knows when to expect and those sorts of things. But I would look at a lot of those strategies are embedded within that Vanderbilt resource. So I would start with that Healthy Bodies Toolkit for girls. And I'll let Wendy talk to.
So everything that Cora said, absolutely. And then some girls are having other issues where they want to regulate their cycles more, or they're having a lot of problems in terms of painful cycles and other things. And so if it's not a matter of just Tylenol or over-the-counter medication, there are other, whether they're birth control, not because you're necessarily using it for contraception, but just be able to regulate the cycles, decrease the amount of flow. And then there are also, and many of you may have used it, medications where you're not having a monthly cycle either. So there are oftentimes pediatric gynecologists and even gynecologists with special needs children, if you go to a large children's hospital, especially, who have experience in terms of helping you make a decision about if you want to use any medication or which medication to use, an injectable medication, a pill, whatever. So there are lots of options available in terms of that. We do try and think about long-term health, though, in terms of long-term heart health, bone health, and also emotional regulation. So it's complicated and not just like I can tell you, just take this Depo-Provera or something like that. So talk to your local doctors. Question.
Hello. Okay. So I have a little bit of a follow-up question about that. my daughter is one of the ones having issues with her menstruation. And our obstetrician gynecologist did mention that because her brain might not be functioning the way a typical brain functions, that could have an effect on how the hormonal contraceptives work. And I'm wondering if you have any feedback on that, because it was something that was interesting to me.
So I'm going to repeat the question, make sure I understood it. I think it's probably for me. But so the question is, because her brain is different, will she respond any differently to the medications that one might use for cycles? So the answer should be no. So there are certain individuals who may have what we call precocious puberty. So they may start going through puberty at a younger age. Sometimes they don't go through full puberty. They may start developing pubic hair or one aspect of things. But sometimes people will see that and then seek an endocrinologist to see if things need to be slowed down so that they don't start going to puberty like at the age of 7 or something like that. But no, in my experience, they respond in the same way to any of the hormonal manipulations for medications.
This was a question from online, and to Dr. Dominguez talk, the individual wanted to know, is 329 the end of the protein, or could there be more? 391, I meant, sorry.
Yeah. For the protein, yes, that is the end, 391. So there are the number of blocks that are formed. However, and we were discussing this earlier, there could be variations in when mutations happen that may make the protein expand. And in fact, it happens in Povins, the other syndrome for Ck2 beta. There is one of the patients actually that because of a splice variant, the ending part of the protein is different. So again, we don't know yet for, let me be, for all of the missense mutants that we have, that's the end of the protein, 391, that's it. But for other mutations that we haven't explored just yet, there could be a potential different ending.
Then we have a question. What is the connection between the brain and the bladder in terms of bladder control?
So I don't know that I know the answer. So I'm going to make a guess and a hypothesis. And if any of the others in the audience want to chime in, they certainly can. So number one is that the brain sends connections to parts of the body. So through the nervous system is sending connections to the muscle, to the intestinal system, to the bladder, to all of those. And in the same way that I talked about low muscle tone in the muscles, so your core or your legs or your arms or things like that, we also see essentially low muscle tone in the intestine and sometimes low muscle tone in the bladder. And sometimes you can have the opposite. You can have spasticity. So you have in terms of the muscle, the nerves sending signals where they're sending kind of too many signals. And so you end up with that. So you can see that both in the sort of in the intestine and in the bladder and that. But I think more of what we're probably seeing in the bladder has to do also with just regulation. So control of that and being able to feel a sense of urgency and when to go and how to hold in terms of those. So I don't know if that was the question or not, but that's at least the biology to the extent that we understand it so far.
Are there any other questions from the audience? We do have some online, so I can.
Thank you. Just a quick question about treatment. Do you think that there will be a treatment for OCNDS within the foreseeable future, meaning 5 to 10 years? And if so, do you think that it would be feasible for it to address the main issues that Dr. Chung showed us before with cognition and speech delay, for example?
I'll start, but I'll say that there are a lot of people in this room that may want to chime in. And I think it's a really important, it's like the central question, right, in terms of thinking about things. So I think you've heard many of us say in terms of treatment, I think there are many ways to think of treatment. And so I think there are treatments, that things we already have available right now, non-medicine. So educational strategy, therapies, a lot of the things Cora has been talking about and others have been talking about through the meeting. Those are already available today. I think our question is, which one of those work best, what frequency, what technique, all of those types of things. There are experiments that the researchers are doing, which are key questions that I think have to do with this question of when. And so there's another question of there's treatment, but when is the treatment most effective? And is there a window of maximal opportunity? And so some of the researchers are doing, we call them reversibility experiments, but being able, I think you've heard this, to be able to see what that window is. We don't know the answer to that question, and it's not, a mouse is not perfect in terms of a person to understand that, but that's another key question that we're trying to answer. When we get into treatment beyond that, and I know some of the scientists were answering this morning, there are treatments that I think of as pills or medicines, and then there are also things that I think of as molecular treatments or genetic therapy or some sort of manipulation in that way. It's not an easy problem to solve. And so I'll say that we need to be able to activate this protein, and being able to do that, just to say, is not, you have to do it in just the right way, and you have to do it in just the right dose. And so it's one of these Goldilocks situations. You can't do too much, you can't do little, you have to get it just right. Now, I don't want to sound skeptical in terms of doing that. The community of medicinal chemists and people that work on this have made amazing strides in terms of being able to see the protein and being able to think about how they can engineer a molecule to fit into that protein and keep it active in just the right way. It's not easy, but I think it's increasingly more feasible to do than it was 5 or 10 years ago. So I'm hopeful in that sense. I'll be very sort of blunt about this. The condition right now is rare. And so when you think about companies that are pharmaceutical companies, people working on this, they simply follow the numbers, right? And so if they can make a decision about, am I going to work on a treatment for Alzheimer's, or am I going to work on a treatment for OCNDS? It's a numbers game for them, right? It's a business. And so they say, what's my potential market? Who's going to be able to benefit from this? And so the reason I say this is, I know you've heard us say this, or at least heard me say it over and over at the meeting. You have to have your numbers counted. And so in doing that, I know Jennifer has the count in terms of the numbers through the foundation, and that's fantastic. The more that they can believe that these are real families with the real diagnoses, know where they are, how old they are, stand up and be counted, even at a very, very basic level. Like, you guys need to have a membership drive in terms of, you've got your numbers counted. You know, it's kind of like get out the vote, right? In terms of getting everyone registered, getting those numbers counted, because it becomes, in the rare disease community, I'll tell you right now, because I see it all the time. Forget the Alzheimer's versus OCNDS. It's Alzheimer's versus, and I'm not trying to make this a competition, but we've got 4 rare disease communities right here. And certain research groups are going around and they're saying, well, should it be HIVE-P2? Should it be, should it be CSNK2A1? I mean, I'm not trying to pit you against each other in any way, but they're thinking about, I've got a hammer, like what conditions or nails that would work with my hammer, and which has the most people. And it's a simple calculation for them. And so the more people that we can get out there, and the more that we know exactly what genetic variations, so we know what hammer, what screwdriver, what wrench is going to work, the more we can partner with them to be able to do that. I do think there are a lot of, within the rare disease space, there are, I call them enabling technologies, but they're essentially these tools of hammers, screwdrivers, wrenches that are being built. And as they're found to be safe and effective, it's mathematically, we would call this a step function. So in other words, it's not as if it's like one by one, very slowly and surely conditions are treatable. It's like it comes in and boom, there's an enabling technology. And so it's not just one condition, it's like 500 conditions that are the nails that fit that hammer, or the screws that fit that screwdriver, right? So we're looking for, how can those enabling technologies be moved forward and move forward in a safe way? And I will say that I won't be surprised if there are some bridges that go from one stage where technology has evolved for one period of time, and they may not be perfect, but they help bridge us to a time where there might be something that's a more permanent treatment or something that is going to be sort of more of a one and done potentially type of strategy, as opposed to something we have to keep doing over and over again. I'll also say that some among you, I'm not sure who it's going to be, but some among you are going to be incredibly generous, brave individuals in your families. That is, with any of these new treatments that need to be tried, we won't know whether they're going to work. I mean, we won't know. We will plan them so that we can do as many safety studies as we can in flies, frogs, worms, fish, mice, other things, to be able to make sure we think they're going to be safe and we think they're going to be effective, but we won't know for sure. And so there are going to be some very brave, generous people who are going to be the first to go forward, and we're going to learn from you, and we're going to iterate in terms of going forward. The exact timeline, which I know is your sort of bottom line, Wendy, give me the answer. Is it tomorrow? Is it a year from now? I wish I knew. But I will say that I'm, for this group, more optimistic when I consider other groups, because number one, your gene, I think, is more druggable. It's something that is more amenable. It's something that what the body continues to need. And so in terms of that window of opportunity, I think the window is probably wider than for some conditions. And I think beyond just a pill, I think there are other strategies in terms of other things to be able to do. So I know that was a long answer. I don't think it's going to be next year. So just to give you in terms of how long the runway is going to be to getting on, I don't think it's going to be within the next 12 months. I think my question is, are we going to be able to make something within the next 5 years? Is it going to be something within the next 10 years? And I think part of this depends on the scientists in this room in terms of them being able to get the funds to keep the engines going, you all in terms of being able to get the vote out and get the numbers, and a little bit of luck with some of these enabling technologies. But I am optimistic that the field is moving forward in a very powerful way. Again, I want to compliment the researchers here that you may not appreciate. They're working together. They're really, really working together. And that wasn't always the way science is done. They're being very generous in terms of, I have to say, even in some cases, their own personal careers, in terms of putting you guys first before them to make sure that they're sharing the data and make sure that they're working together. And they're not sort of glory hogs where they're taking all the credit. They're putting you guys first in terms of the way they're approaching this. And that's to their credit, that they are not, they're being incredibly professionally magnanimous to make sure that you guys benefit.
So just for the scientists, from the scientists that are here, we cannot give you an answer to the time. So the way we are thinking about it is the first thing we need to do is to figure out for each mutation whether it increases or decreases or changes somehow the activity or the location in the cell of the protein of the new CK2. So that's the first thing that we need to figure out. And Heike has done tremendous work, and you have to thank her because she's been working on this for the past 3 years. And she already has tested, I think, 18 mutants, if I recall properly, in her laboratory, and found that all of those mutants decrease the activity. So now we have to find something that increases that activity or other strategies. So, and following up with this, Jenny in Germany is now proposing a grant to do a high throughput screening from new compounds that could elevate the activity of CK2, thinking for all of those mutations that we already know that have decreased activity, can we find something that stimulates the activity? So again, we may find other mutations that increase the activity and we need to decrease it. And we may find other mutations that go places in the cell where they should not be, and we need to get rid of them from there. So the more we know about the mutations, the more we know about, how do you say that? The mutations, the more we will be able to do. If we don't know that, if we don't know the mutations, we cannot help. And again, we cannot tell you time, but we are all trying together. Karsten and Jenny also are doing crystallography studies. We have Joachim over there doing binding and trying to figure out also activity and who binds to each other. I mean, we have cilia, cellular functions. So we are all working together to figure out if the mutants, again, decrease, increase, or change somehow. And then that's the only way to find a targeted therapy for the patients.
I think that we could ask questions all day long, and I can't thank you guys enough for your time and your dedication, and also for everyone here today for your thoughtful questions and online as well. We are going to have a follow-up Q&A in about a month or so from now. So that way everyone can kind of digest everything they've heard today. People online have a chance to watch these at their leisure. And so we'll have an hour Q&A where virtually we'll ask the online questions and any questions that were left unanswered here today, which will be fantastic. Thank you for giving your time to do that for us.